In this video, you will learn about the processes that occur in order to go from DNA to you. In detail, we will explain DNA structure, transcription, RNA editing, translation, protein folding, and the importance this all has to humans. This is the structure of a piece of DNA in a one-dimensional model. DNA is in the form of a double helix, meaning that it has two strands spiraling away from each other. The monomers of DNA are called nucleotides, which consist of a phosphate group, a deoxyribose sugar, and a nitrogenous base. Each phosphate group connects to a deoxyribose sugar, and each sugar connects to a nitrogenous base. A nitrogenous base is either adenine, thymine, cytosine, or guanine. Adenine always pairs with thymine, and this pair always contains a double hydrogen bond. Cytosine always pairs with guanine and always contains a triple hydrogen bond. Most DNA is different and contains different patterns or orders of nitrogenous bases. DNA is also anti-parallel, which means the helices are parallel but also move in opposite directions.
In addition to gene transcription, there is also an important editing phase. During this editing, unwanted parts of the gene called introns are removed from the pre-RNA by a process called splicing. The remaining parts, called exons, stay in the RNA, now called messenger RNA. The exons will leave the nucleus and be used in the next step of protein synthesis. The first part of protein synthesis is transcription, which is the way a segment of DNA is copied into pre-RNA by the enzyme RNA polymerase and is then edited into mRNA. The process of transcription begins at promoter regions near the beginning of a gene. At the end of a gene, there are termination sequences, which tell the RNA polymerase when to stop transcribing. The RNA polymerase unzips the DNA, then a pre-RNA is created with the corresponding nitrogenous bases on one side of the helix. This creates a copy on the other side of the helix. However, there are some differences between DNA and mRNA. mRNA does not contain the nitrogenous base thymine. Instead, it has one called uracil. RNA is also in the shape of a single helix, unlike DNA's double helix shape.
Translation is the next step in protein synthesis. This process is when the mRNA is read by a ribosome, which is an organelle made of proteins and RNA, to produce the correct amino acids for a growing polypeptide chain. The ribosome reads the mRNA strands in groups of 3 bases at a time, called codons. Codons call for a specific amino acid or a start-stop. The start-stop codons tell the ribosome when to start and stop, calling for amino acids to build to the chain. Another type of RNA, called transfer RNA, picks up an amino acid from the cell and brings it to the ribosome. The tRNA knows which amino acid to bring because it has an anticodon, which has the opposite bases from a codon. The anticodon and the codon click together and the amino acid is taken by the ribosome. This process repeats until there is a long chain of amino acids called a polypeptide chain. This polypeptide chain eventually breaks off and makes a protein.
There are 4 levels of protein structure organization. The primary protein structure is simply a sequence of amino acids which are chosen and delivered by the translation process. This is a one-dimensional structure. The secondary protein structure occurs when the sequence of amino acids are linked by hydrogen bonds. This forms the two-dimensional protein structure, including alpha helices and beta pleated sheets. The tertiary protein structure is when alpha helices and beta pleated sheets attract and connect, and the protein folds. This structure is three-dimensional. The quaternary structure of protein includes more than one amino acid chain and is also three-dimensional.
DNA to RNA to protein to you is often called the central dogma of biology. But why is this important? Well, eventually, proteins are made in this process. But the proteins make up you. But why do the proteins matter? Well, the proteins are the monomers of many things in your body, including bones, muscles, insulin, cartilage, skin, hair, and blood. Proteins also help carry out certain functions in the body, such as transporting materials throughout the body. Remember, without proteins, there is no you.